Hello children, welcome to biology class. We are discussing the lesson locomotion and movement. Last class we discussed about the structure of skeletal muscle. Hope you all studied it well. Now let's discuss about structure of contractile proteins. Yesterday we had discussed about two contractile proteins in skeletal muscles. Which are they? Actin and myosin. Okay. Now let's discuss the structure in detail about the structure of these two proteins and the mechanism of muscle contraction. Okay. So for first is about the structure of contractile proteins. You observe this diagram. This is actin filament. This is myosin filament. Each actin or thin filament is made up of two filamentous actins helically wound to each other. The F-actin, that means the filamentous actin, is a polymer of monomeric globular actions. Two filaments of another protein, tropomyosin, also run close to the F-actins throughout its length. So a complex protein, troponin, is distributed. Here you can see troponin, tropomyosin, and this F-actin. Okay, can you observe? So, a complex protein troponin is distributed at regular intervals on tropomyosin. So, in the resting state, a subunit of troponin masks the active binding site of myosin on the actin filament. Okay, so that is about actin filament. Now, let's discuss about myosin. It is also a polymerized protein. Many monomeric proteins called meromyosins constitute one thick filament. So each meromyosin has two important parts. Which are they? A globular head. You can see it here. A globular head with a short arm and a tail. The former being called the heavy meromyosin, HMM, and the later the light meromyosin, LMM. The HMM, that is the heavy one, is a head and short arm projects outwards at regular distance and angle from each other from the surface of a polymerized myosin filament and is known as crow's arm. The globular head is an active ATPase enzyme and has binding sites for ATP and active sites for actin. The ATP is required for doing the action. See children, actin filament with troponin and tropomyosin, and myosin filament, head, stalk and hinge or tail, short arm and head. Clear? Now let's discuss about the mechanism of muscle contraction. It is explained by sliding filament theory, which states that the contraction of muscle fiber takes place by the sliding of the thin filament over a thick filament. Okay, so this diagram is there in our textbook on page number 307. You observe the diagram. You observe this diagram. Okay. So, the mechanism of muscle contraction is best explained by sliding effect. See, can see sliding filaments here. Okay, so this is explained by the sliding filament theory, which states that contraction of a muscle fiber takes place by the sliding of a thin filament over a thick filament. Clear? As you can see, thin and thick filaments here. So, sliding of thin filament over a thick filament results the muscle contraction. Okay, the contraction and relaxation. Now, we are going to explain the mechanism behind that. Muscle contraction is initiated by a signal sent by the central nervous system via motor neuron. So, motor neuron along with muscle fiber connected to it constitute a motor unit. Okay. So, the junction between a motor neuron and the sarcolemma of the muscle fiber is called neuromuscular junction or motor end plate. So, a neural signal reaching this junction releases a neurotransmitter. That is acetylcholine, which generates an action potential in the sarcolemma. So what happened? This spreads through the muscle fiber and causes the release of calcium ions. You observe this one, children. they release calcium ions into the sarcoplasm. So increased calcium level lead to binding of this calcium with the a subunit of troponin on actin filaments, and thereby remove the masking of active sites for myosin. So what happens during that time? Utilizing the energy from ATP hydrolysis, the myosin head, you can see here the myosin head now binds to the exposed active sites on actin to form a cross bridge. Okay, so this pulls the attached actin filament towards the center of A band. Then what happens? The Z line attached to these actions are also pulled inwards, thereby causing a shortening of the sarcomere. That means contraction. Shortening means contraction. So children, it is clear from the above steps that during shortening of the muscle, that means during contraction, the I bands get reduced, where the A bands retain the length. So the myosin releasing the ADP and inorganic phosphate goes back to its relaxed state. A new ATP binds and cross bridge is broken. The ATP is again hydrolyzed by the myosin head, and cycle of cross bridge formation and breakage is repeated, causing further sliding. So this process continues till calcium 2 plus ions are pumped back to the sarcoplasmic cisternae, resulting in the masking of actin filament. This causes the return of set lines back to the original position. That means relaxation. The reaction time of the fiber can vary in different muscles. Repeated activation of the muscles can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. You might have heard about muscle fatigue and all. So muscle contain a red colored oxygen storing pigment, that is myoglobin. Myoglobin content is very high in some of the muscles. So such muscles are called red fibers, which gives a reddish appearance. These muscles also contain plenty of mitochondria, which can utilize a large amount of oxygen stored in them for ATP production. So these muscles therefore can also called aerobic muscles. On the other hand, some of the muscle possesses very less quantity of myoglobin and therefore appear pale or whitish. These are white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on anaerobic process for energy. Okay, so that is about the skeletal muscle contraction. So in short, how can we write myosin head hydrolyze ATP and become reoriented and energized? Then myosin heads bind to actin, forming cross bridges. Myosin heads rotate towards the center of sarcomere. Before that, you have to mention the calcium release. That means the release of neurotransmitter acetylcholine. Okay. Then step 4, as myosin head bind to ATP, the cross bridge detach from actin. Okay. So while explaining it or while reading the theory part, you have to observe this diagram too. It will be very helpful. Okay, children. So that's about mechanism of muscle contraction. So from this lesson we have to discuss only these topics. Okay, and the remaining topics in the textbooks are omitted by CPSC according to the revised syllabus. So only few topics are there in this lesson, right? Only the structure of skeletal muscle and mechanism of muscle contraction. So study thoroughly, children. Okay, you read the textbook along with the diagram. You open the textbook and you observe the diagram, then you study each and every points. Okay, so study well, children. Thank you.